Welcome back, dear students. You know that we are discussing asexual reproduction strategies in bacteria. In the previous part, we discussed three, and today we will start with the fourth, and it is cysts. Cysts जब हम डिफाइन करते हैं उससे पहले पता होना चाहिए हमें कि असल में ये क्या है और किन में पाए जाते हैं. ठीक है. These are the resting cells. Resting cells means those cells which help the bacteria to rest during unfavorable conditions. Okay, and these are found in the genus Azotobacter. Now, cyst. What is a cyst? Cyst is actually when the entire bacterial cell it gets enveloped by additional protective layers. The whole structure, I mean the bacterial cell, along with the additional protective layers, this whole structure is known as cyst. Okay. And cysts are known to act as perinating structures. Perinating structures means these help the bacteria to survive during unfavorable environmental conditions, or you can say these help the bacteria to overtide unfavorable conditions. And these cysts they are resistant to desiccation. That means if there is loss of water from the medium, the cyst it remains viable. Okay, but it is not resistant to heat. If you will give a heat treatment to cyst, it will lose its viability. Now, endospores. Endospores are the perinating structures just like cysts, and these are found in some large gram-positive bacilli. Usually, they are found in large gram-positive bacilli, I mean rod-shaped bacteria, and they have also been found in few cocci and spirilla. Example is Sarcina lutea and desulfivibrio desulfuricans. Okay. Endospores are intracellular resting cells. They are formed within a vegetative cell called sporangium. Unlike cysts, endospore formation involves only a part of the original cell. जैसा कि मैंने आपको बोला, जो cysts होते हैं उनमें entire bacterial cell gets converted into the cyst, while as in case of these endospores, a part of the bacterial cell it becomes organized to form a spore which is known as endospore. Why this is known as endospore? Because it forms inside the bacterial cell. Now the shape of the endospore. Its shape is variable and can either be oval or spherical. It can be either oval or spherical. I mean the shape of endospore which is present inside the bacterial cell, it can be either oval or spherical. Now position within the parent bacterial cell, it can be either central or subterminal or terminal. Let's make it more clear. When we say central, it means it is located here. When we say subterminal, it means it is located here. And when we talk of terminal position, it means it is located here at the end. Now, formation of endospore. First of all, what are the conditions essential for the formation of endospores? Endospores are formed during the stationary phase of the cell. Stationary phase means when cell is not dividing. Cell doesn't divide when there is scarcity of nutrients in the medium. Okay, so you have to simply remember that endospores are formed during the stationary phase of the cell when it is not dividing due to lack of nutrients in the medium. The process of sporulation, I mean spore formation, involves two nucleids inside the vegetative cell coalesce. Coalesce means join. Okay, they join to form the axial chromatin thread. It will get more clear on the next slide when you will see the image. Okay, a transverse septum then develops at one pole of the cell, और इससे क्या हो जाता है? It delimits a small daughter cell which is called as spore primordium from the larger cell. Okay, the septum does not develop into a wall. Instead, the membrane of larger cell grows around and envelops the spore primordium. Okay, this is the third step. So the first one is two nucleids are there. They coalesce, they join to form the axial chromatin thread. The second is a transverse septum develops at one pole of the cell. At one end of the cell, a transverse septum is formed, and this delimits a small daughter cell. It forms a small daughter cell at the pole. Okay, and then the septum does not develop into a wall. इस septum पे एक wall नहीं बनता है. Okay, instead the membrane of larger cell grows around. It grows around the smaller cell, and uh, the smaller cell then acts as spore primordium. Okay, then spore primordium increases in size and matures into a spore. It becomes opaque and refractive. Okay, when spore becomes mature, it becomes opaque and it gets the feature of refractivity. Okay, it becomes refractive. It becomes able to refract light. Okay, this is the indication that spore has become mature. And this process of spore maturation, I mean the formation of spore inside the bacterial cell, this is completed in a span of 18 to 20 hours. It takes time of 18 to 20 hours to form a fully mature spore. Then the liberation of endospore is mediated by autolysis of the sporangium. जिस सेल में spores बने होते हैं, then later on it lyses. It is walls break down and the spore endospore is liberated. Here you can see this is the vegetative cell. Here at the top you can see the two nuclei, they coalesce to form the axial chromatin thread. Okay, then the third step is a transverse septum forms here, and this septum does not it is not followed by wall formation. Instead, the membrane of the larger cell, it envelopes this smaller cell, and this smaller cell becomes encased. Okay, it becomes encased, I mean it has its own cover and acts as the spore primordium. ये फिर function करता है spore primordium, and this spore primordium later on increases in size and ultimately it matures. Okay, then after maturation there is lysis, there is breakdown of the wall. You can here see there is breakdown of the wall of this parent cell. Okay, and the endospore is later on liberated. I will repeat these things. First of all, you have a vegetative cell. The two nucleides of the vegetative cell, they coalesce and form the axial chromatin thread. Then a transverse septum is formed here. This is not followed by wall formation. Instead, the membrane of the larger cell, it envelopes the smaller cell here, and this smaller cell becomes encased within the larger cell. Okay, this smaller cell is known as spore primordium, endospore primordium you can say. And ये जो endospore primordium होता है, later on it increases in size, and ultimately when it becomes mature, the wall of the parent cell, it lyses, it breaks down, and the endospore is liberated. Now the structure of endospore. What is the structure of endospore? It consists of following components from inside out. अगर हम अंदर से बाहर की ओर देखेंगे तो इसमें 4 components है. The first one is core. It is a central protoplast, and it is mainly composed of DNA, tRNAs, ribosomes, enzymes, and accessory factors. ठीक है? And it is enclosed by a thin membrane which is called as core membrane or inner membrane. So 4 components है एक एंडोस्पोर के. अगर हम अंदर से बाहर की ओर देखेंगे तो पहला वाला जो कंपोनेंट है उसको हम बोलते हैं कोर. ओके, द सेकंड वन इज कॉर्टेक्स. इट इज़ मल्टीलेयर्ड रीजन अराउंड द कोर. कोर से बाहर जो रीजन होता है उसको हम बोलते हैं कॉर्टेक्स, एंड इट इज़ लार्जली कंपोज्ड ऑफ कैल्शियम डाइपिकोलिनिक एसिड पेप्टिडोग्लाइकन कॉम्प्लेक्स. ओके, इट इज़ कंपोज्ड ऑफ कैल्शियम डाइपिकोलिनिक एसिड पेप्टाइडोग्लाइकन कॉम्प्लेक्स. याद रखना बेटा. Then the third one is spore coat. It is an electron dense layer outside the cortex. फिर cortex के बाद एक और लेयर है जो electron dense layer है जिसको हम बोलते हैं spore coat. And it is differentiated into inner coat and an outer coat. It is further divided into an inner coat and an outer coat. And this spore coat is composed of proteins. Around 80% of the proteins which are present in endospores, they are present in the spore coat. Okay, and accounts for 30 to 60% of dry weight of the spore. जो spore coat होता है, the constituents of the spore coat, they account for 30 to 60% of the dry weight of the spore. And finally, the exosporium. It is the additional, or you can say outermost covering, but it is not found in case of all the bacteria. It is found in some bacteria. Example, Bacillus cereus. And it is composed of polysaccharides, proteins, and some lipids. याद रखना, exosporium is the additional, or you can say outermost covering in some bacteria. In some, it's not present in all bacteria. Example is Bacillus cereus, and this exosporium is composed of polysaccharides, proteins, and some lipids. Okay, here you can see this is the diagrammatic representation of the structure of spore, endospore. This is the parent cell, or you can say mother cell, and this mother cell is known as sporangium because it produces inside an endospore. Okay, the endospore structure is such that you have the core here, then core के बाद क्या आता है? You have here the cortex. Okay, you have here cortex. We have already discussed the details about cortex. Cortex के बाद आता है spore coat, this green ring, and finally after spore coat we have here the exosporium. Okay, and remember, exosporium is not found in case of all the bacteria. I will repeat these things. This is the core. After core we have this cortex. Okay, calcium dipicolinic acid peptidoglycan complex. Okay, it is composed of calcium dipicolinic acid peptidoglycan complex. Then this is the another layer which is known as spore coat. Okay, and finally after the spore coat we have here this exosporium. Okay, now germination of endospore. How endospores germinate? The free endospores that are released from the parent cells, or you can say sporangium, they have undetectable metabolism. आप ये नहीं देख पाओगे कि ये मेटाबॉलिज्म दिखा रहे हैं कि नहीं दिखा रहे हैं, ठीक है, but they retain the potential capacity to germinate and develop into vegetative cells. Although they are dormant, but they have the capacity, they have the power to develop into the vegetative cells, to develop into the bacterial cells after germination. Okay, they germinate under favorable conditions. Favorable conditions means when there is sufficient nutrition available in the environment, when there is optimum humidity, when there is stable temperature. Okay, under favorable set of conditions these endospores they germinate. Okay, and germination is completed in 3 stages. The first one is imbibition of water and consequent swelling. For example, this is an endospore, it imbibes water and this swells. Okay, it swells. After this first stage, the endospore, it loses its refractivity, it becomes somewhat translucent, it's not that much opaque. अभी ये opaque नहीं है. Okay, इसकी opaqueness कम हो गई, and because of this it loses its refractivity. Then finally rupture of spore coat and the growth of new vegetative cell. फिर क्या होता है बाद में? जो spore coat होता है it ruptures, फट जाता है. Okay, and finally the growth of new vegetative cell begins. Why endospores? What is the need for the formation of endospores? Why bacteria produce endospores? Bacteria produce endospores for a number of reasons, and these are highlighted here. They confer survival value to tide over unfavorable conditions. जो bacteria होते हैं, okay, at some point of time they face unfavorable conditions, and during those unfavorable conditions they produce the endospores which are able to resist those unfavorable conditions. Okay, they provide resistance against heat. Okay, UV and ionizing radiations, many toxic chemicals like hydrogen peroxide, formaldehyde and other biocides. Okay, so excuse me, they provide resistance against heat, and endospores of some species of bacillus have been found to tolerate a temperature of 150 to 170 degrees Celsius. Okay, excuse me. They can also withstand UV and ionizing radiations and many other chemicals like hydrogen cyanide, formaldehyde, and other biocides, their effect can also be nullified by the endospores. Okay. Now some important remarks about endospores. The resistance of endospores is attributed to calcium dipicolinic acid complex. जो एंडोस्पोर्स में रेजिस्टेंस होती है अगेंस्ट हीट, अगेंस्ट यूवी रेडिएशंस, अगेंस्ट हाइड्रोजन peroxide, formaldehyde and other biocides, it is speculated to be due to the presence of कैल्शियम डाइपिकोलिनिक एसिड कॉम्प्लेक्स, जो हमने डिस्कस किया था स्ट्रक्चर में. Only one endospore is formed per cell. That means it is not a means of multiplication. इससे बैक्टीरिया का नंबर इंक्रीज नहीं होता है, बल्कि पेरिनेशन होती है. इट हेल्प्स द बैक्टीरिया टू पेरिनेट, टू ओवरटाइड द अनफेवरेबल कंडीशन. याद रखना, एंडोस्पोर्स आर नॉट मेंट फॉर बैक्टीरियल मल्टीप्लिकेशन. फॉर मल्टीप्लिकेशन ऑफ बैक्टीरिया देयर इज बाइनरी fission. Okay, endospore formation mainly occurs as a consequence of unfavorable conditions. Okay, so that the bacterial cells, the bacteria, it can overtide the unfavorable conditions. This is all about the endospores and the cysts. I hope you all understand this. See you soon with a new lesson. Till then, stay blessed. Thank you.